그치가.
Hey guys, good afternoon and welcome everybody to a wonderful session of Geotalk today. We have Professor Jennifer Botha, who is currently the director of Center of Paleosciences at the ESI, which is also known as GENUS. Her research emphasizes on the aspects of paleobiology, paleoecology, and the focus of her research mostly is around the aspect of life history, responses of extinct vertebrates to environmental change. I promise her that I will make sure that I don't leave any aspects which I was taught about in the bio. So she also emphasizes the aspects of Permian mass extinction and the early Triassic mass extinctions. So the techniques that she uses are mostly biostratigraphy, morphology, osteohistology to test their theories on species survival during mass extinction events. And our special interest revolves around osteohistology, fossil bone microstructure that provides novel information about life history of extinct vertebrates. And now you might wonder why this is all important to us today. This is a point where Jennifer's research is very interesting. In today's modern world, we are going through another mass extinction, which is the sixth one in the background. And her research is relevant in all of these aspects, and she will unfold all of this through the talk that she's highlighted here, Apocalypse Then, the greatest mass extinction. Over to you, Jennifer.
Hi, good afternoon, everybody.
Okay, thanks everyone. Yeah. We are back. Jagamoy, thank you for that introduction. Are there any questions? Okay, Jen, over to you.
Again, good afternoon, everybody. Thank you for having me here today. I've only recently been in Johannesburg just for about 8 months now. But this is the first talk I've given at Wits. So thank you very much for having me today. This is an All right, so today I'm going to be talking about the end Permian mass extinction. Now, there have been 5 mass extinctions in Earth's history, but the end Permian mass extinction is fondly nicknamed the mother of these mass extinctions because it changed the composition and structure of both marine and terrestrial ecosystems so substantially that it's considered to be the most catastrophic biotic crisis in Earth's history. Now, the marine extinction has been very well studied and well dated. The latest studies suggest that it lies between 251.9 and 251.8 million years ago. Now, along with these radiometric dates, there have been many studies about the organisms. So we have seen the extinction of organisms and the appearance of new taxa. We have seen sea level changes, increases in the ocean temperature, and marine anoxia and eoxinia. Now, probably for many decades, researchers have debated as to what was the major cause for the extinction. And there's been much going back and forth. At one stage, there was even a suggestion of something similar to the end Cretaceous extinction of a bolide impact, that a meteor had crashed into the Earth. But most researchers today agree that it is the Siberian traps. Now, the Siberian traps today is a large igneous province in what is today Siberia, Russia, and it represents some of the largest volcanic eruptions in Earth's history. And it lasted at full intensity for about 1 million years, and it coincides with the timing of the end Permian mass extinction. So, how could volcanic eruptions cause such a massive extinction? So for those of you who don't know that much about volcanic eruptions, but now think about this in terms of effects lasting for millions of years, the lava and pyroclastic flows would have affected the immediate area. And remember that province there is actually really large. But it's the sulfur and the ash that would have blocked the sunlight. And these effects can last for hundreds and thousands of years. And it's the greenhouse gases. Now, I'm sure you are familiar with the term greenhouse gases, runaway greenhouse effect. I'm sure you have heard about that when we have discussed today's mass extinction, the 6th mass extinction that we're currently going through with the term global warming. So carbon dioxide, methane, and water vapor are all greenhouse gases, and they have the ability to increase global temperatures. And it doesn't take much. It takes 1 or 2 degrees for organisms to start getting really uncomfortable and some of them to really start to battle to adapt to these changing conditions. And these effects can last for millions of years. And then the chlorine and fluorine gases that also come out of these volcanic eruptions destroy the protective ozone layer, and they allow harmful UV rays to penetrate. So when you're living at this time, it's really not a great time to live. Now, as I said, most researchers have concentrated on the marine aspect of the extinction. That is because many thought that there were no complete terrestrial Permo-Triassic boundary sites in the world. But over the last 3 decades, we have found sites in South Africa, in Australia, Antarctica, Russia, and China. So we really can examine the timing and mechanisms of the terrestrial aspect of this extinction now. So we have found evidence of climate change. We look at the sedimentology, and we have found this evidence at several sites around the world, including South Africa, Antarctica, Australia, Spain, and Russia. And we see a change from, you can see these big winding rivers here, high sinuosity meandering rivers with expansive wet lowland floodplains. Apologies, everyone. It's an alarm. It's not actually my phone. And this changed through a straightening transition change to braided ephemeral rivers with dry floodplains and flash floods. Now, associated with this is a negative carbon-13 excursion. So here we have the Meishan section. This is the typical the strata type section in China, and you can see this curve here, this blue curve with this huge excursion here, this negative excursion, and then it carries on here. So with the marine extinction, it is associated with this negative curve. So people have looked for similar curves in the terrestrial sites. And researchers have looked at Australia, we have looked at sites in South Africa, Antarctica, and China, and we have found similar curves where we have found a negative carbon-13 excursion. And this really represents the very beginning of a whole lot of early Triassic carbon-13 excursions. And it represents massive carbon cycle fluctuations, extreme climate perturbations. So basically really, really bad environmental stress. So you can expect to see effects on the plants and animals during the early Triassic. Now, the marine biota have been very well studied, but the rapidity and the extent of biodiversity loss in the terrestrial realm is still under debate. For example, this is a study by NOAC that was done in 2019. They looked at macrofossils of plants, and they decided that there was no major decrease in plants. This is the end of the Permian, this line here. There was no major decrease across the Permo-Triassic boundary. So they did not think that there was a major extinction amongst the plants. However, and this is actually quite funny because it came out in the same edition, both of these papers, this is Fielding in 2019 and the other one, NOAC, came out both in Nature in the same edition, one saying that there was no real mass extinction amongst the plants, the other one saying there was a drastic extinction amongst the plants, which just shows you the lack of consensus that we have at the moment. This study used palynology though, this is using pollen, and found that there was a disappearance of the gymnosperm dominated flora, and they were replaced by the leucopsid dominated flora here. So you can see there's still much work to be done. Okay, but what about the animals? And this is where I come in. So I just need to express to you that South Africa is very, very, very special. It preserves the best preserved terrestrial Permo-Triassic boundary site in the entire world. So we have been able to look at infinite details, basically under a microscopic lens. So we've looked at the biostratigraphy, the lithostratigraphy, we've looked at geochemistry. Recently, we've begun radiometric dating, and we've really been able to look at the biology, even the life history, and the changes that the animals have gone through during this extinction. And what we found is that it's quite a complex scenario of the animals going through this extinction. For example, the Parasaurian parareptiles go completely extinct. This is a group, parareptiles are the sister taxon to true reptiles, to diapsid reptiles. So they are all extinct today. And the Gorgonopsians, these are very large carnivorous therapsids. Now I'm going to say therapsids quite often, so I'd like you to get used to it. These are the ancestors of mammals. And these Gorgonopsians were the most dominant predators of the late Permian. They are these saber-toothed type predators that I'll show you in a moment. And then there are other clades, such as the Animodonts and Therocophalians that did survive the extinction, but at reduced diversity and abundance. And eventually they went extinct as well. However, what is interesting is that some clades did survive the extinction and went on to become highly diverse and abundant and form very important parts of the early Triassic post-extinction environment. So you can see here the procolophonid parareptiles. They became quite diverse for a little while, but then they petered out. But the temnospondyle amphibians did very well for a time. These are the diapsid reptiles that gave rise to archosaurs, which are dinosaurs, Okay, and the cynodonts, another very important group, because the cynodonts are the direct ancestors of mammals. If they had not survived this extinction, we wouldn't be here today, and I wouldn't be talking to you today. So this is what I usually do. I am not a geologist. I'll say that up front. I work with many clever people that are experts in geology, in geochemistry, in sedimentology, etc. I like to work on the biology of the animals associated with the extinctions to see who will survive, who will go extinct, and why. What are the survival strategies? So I just wanted to show you an example of the kinds of things that we do. So my colleagues and I were looking at the early Triassic pattern, and we looked at what has already been mentioned, fossil bone microstructure. Now, just to explain to you what that is, we'll take a sliver of fossil bone and we'll grind it down until you can see light through it and put underneath the microscope. And the positions of all the blood cells, the bone cells, and the collagen fibers all remain in that same position as it was in life. All the organic components have disappeared, but the positions remain the same. And we can tell all sorts of different things about the life history of an extinct animal this way. So we looked at that, and we looked at the body sizes of these therapsids going through this extinction, and we did some modeling, and we found that the animals that were least likely to go extinct were the ones that had a truncated development. Now, this means that they bred early. So I could tell from the bone microstructure that these animals were not living for very long. They were not reaching adulthood. So, for example, Lystrosaurus, and I'll say him quite often, he's a very abundant dicynodont therapsid, a herbivorous therapsid that was extremely abundant during the early Triassic, and much work has been done on him to understand why he survived, but not only survived, became very abundant during these very harsh conditions. And if we look at Lystrosaurus, even though thousands is about 3,500 to about 4,000 specimens all over the world of Lystrosaurus, not one of them is an adult, is fully grown. But these animals were breeding. So they were breeding younger, and they were dying younger. And we have taxonomic evidence to support this. So I want to just show you, this is a very large bone bed here consisting of several Lystrosaurus individuals. These pink spots here indicate skulls. So this is just a jumble of disarticulated skeletons. You can see it's a real mess here. But amongst the disarticulated elements, we see some articulation, such as these ribs over here. So I want to come back to those ribs in a moment. That's why I want to point them out in this picture, because we can use that for evidence of avidity, which I'll tell you about in a moment. But here we can see numerous specimens in a single bone bed, and we find several bone beds of Lystrosaurus. Now, although we will find aggregations of other species to some extent, we will never find it to the same extent as we find with Lystrosaurus. Lystrosaurus was very abundant, so it was successful. It was doing something that allowed it to survive. So what we see is the disappearance of Permian taxa. So the dicynodonts, these are the herbivores. You can think of them as the cows of the Permo-Triassic. And then the Gorgonopsians, you can think of these as the big carnivores that ate the dicynodonts. And then we do see some survivors. So we do see taxa going through the boundary, and this includes Lystrosaurus, but different species. We have 4 different species of Lystrosaurus in this country. And then we see the appearance of, here are 2 new species of Lystrosaurus and other Triassic taxa, including Proterosuchus. He's quite important because he's found just above the boundary. And Proterosuchus is an archosauriform, which is the precursor to archosaurs, which include dinosaurs. Now, I'm not going to go into detail in all of this. All I really want to point out here is that we have been to many different sites around the South African Karoo Basin. And what we are starting to find is that there is a similarity in the facies associations at these different sites. So I'm not going to go into detail about the A to C here. What I want to show you is that we are finding A, B, C, D, E at all of these sites. We don't find them as much here at Fricacus Corp because this is in the northern part of the basin. It's over here. And this is where the sediments start to thin out. So we don't have as much preservation here, and we do not have a complete perma-Triassic boundary here either. But where we do have complete ones, we find very similar sediments. And we find these boundary beds, which I'll show you in a moment, all the way through every section that we have logged at the various different sites. And these indicate a progressive lowering of regional groundwater, increased climatic drying, and very unpredictable rainfall. Now, one of my colleagues, Roger Smith, noticed that amongst all of these taxa going through the boundary, there were three pulses, three extinction pulses. The first one just below in the occurred here, where most of the dicynodons went extinct, of the Gorgonopsians. And then we have a survivor taxa that did survive, didn't last for very long. They actually all went extinct very close to the boundary. So they just managed to peek over the boundary, but then they couldn't handle the early Triassic and they died off. And this pattern of a Glossopteris dominated flora and the appearance of Dichordia dominated flora. So what do we look for when we are looking for the many things, not just one thing, but the first thing we look for is a sedimentological marker. And these are in the form of rhythmically bedded reddish brown. Yeah, you can see the reddish brown here at the lower part here. This is a nice clear section. And olive gray, here are the olive gray, siltstone, mudstone couplets. They can range in thickness anywhere from 3 meters to 7 meters. It doesn't really matter. But these mudstone, siltstone couplets really do cover the perma-Triassic boundary or that transition of the boundary beds. Here's another section at Bethel. This is also near Batuli. Both of these sites are in the Free State. So above those boundary beds, we see, as I mentioned, the appearance of three very important taxa. So the first thing we look for when we are looking for Triassic strata are the two different Lystrosaurus species and Proterosuchus. Now, we've also found a negative carbon-13 excursion at many of the sites in South Africa. This is where we see changes in community structure. Now, this is not the end of the story. There are still many debates flying around concerning the perma-Triassic boundary in South Africa. And perhaps the biggest one currently is, where is the perma-Triassic boundary? So there have been debates in the literature as to the timing of the perma-Triassic boundary. Is it the same extinction as the marine extinction? So we are looking at the terrestrial extinction and the marine extinction. Now, if they were happening at different times, it would suggest that there are two different extinctions. So if this is incorrect, where we have placed the perma-Triassic boundary for so many years, and it is not at a similar date to the marine extinction, you would see two different extinctions, and this has implications for the faunal and floral extinctions. And researchers have cast doubt on the plant and animal extinctions. And there's also been doubt about whether the climate changed to very dry, whether there really was aridification. So most researchers agree that there was an increase in temperature, but some think that it became hot and wet, and some people think it became hot and dry. So one of these researchers is Bob Castaldo, and a couple of years ago, he retrieved a radiometric date from the South African Karoo. He retrieved it in Permian strata at 253.5 million years. Now, if you remember, the marine extinction is dated at about 251.9 million years. So this is definitely Permian. And he used paleomagnetostratigraphy and sedimentation accumulation rates to suggest that where we had traditionally placed PTB was far too low, and that it should be placed either at the base of the Catberg Formation, which is traditionally Triassic. So this is the Catberg Formation here, and the Palenkloof member has always been Permo-Triassic, or even perhaps somewhere within the Catberg Formation. Now, the implications for this is that you would get a Permian Catberg Formation, and there are implications here in terms of the extinction, because we have an extinction here going all the way through. So is this all Permian, and are the dates completely different to the marine extinction? So going back to the plants again, the study by Nowak, where he looked at macrofossils. And I do need to remind you that macrofossils are quite rare at Permo-Triassic boundary sites, which is a bit of a pity, but it means we have small sample sizes. So although they did not find a drastic decrease in plant diversity across the Permo-Triassic, I still have to wonder, is this a true scenario, because we find so few macrofossils. But Bob Castaldo, during his research here in the Karoo Basin, he found Glossopteris above where he placed the Permo-Triassic boundary. So he noted that Glossopteris had been found in the Catberg Formation. I do want to point out, I'm sure he did find Glossopteris in the Triassic, I don't doubt that. But this is after a publication by Ward et al. In 2005, and they never published where the boundary was. So I don't actually know where this line comes from. The line here is correct, but in terms of his log, I don't know where. I don't know if that's correct. But even if Glossopteris did survive, that doesn't mean we change the boundary, or that suddenly this whole Catberg Formation is Permian. It means that there were some species of Glossopteris that survived, because the Glossopteris, the whole flora of Glossopteris consists of many, many, many different species. I also need to point out that this was done at a site called Old Lewitsburg Pass. My colleagues and I do not look at this site at all. There is a fault that runs through this pass, and it makes life very difficult to try and figure out what's going on, because you will find Permian taxa right next to Triassic taxa. So how do you figure out the biostratigraphy? And if things have shifted, what happens to the paleomag? What happens to the geochemistry? Where are you with the radiometric dates? It's a very, very, very difficult site to work with and to know precisely what is going on in that column. So I want to bring you to a really nice site. This is my baby, Noordgedacht 68. My students have been there already. I just call it that for short. It actually runs over 3 different farms, and it consists of a hill called Loskop and a butte called Spitzkop. They're about 800 meters apart, just north of Betuli in the Free State. This is what the hills look like. This is Loskop here on top and Spitzkop. And we consider them to be equivalent in age due to the lateral continuity, the continuous sedimentation, and similar faunal assemblages. So now to give you some geology, because that is what you guys do, just to make you happy. So what we see on the lower slopes of Spitzkop, for example, we see alternating reddish-brown mudstone and olive-grey siltstone interspersed with fine-grained sandstone bodies. We see these calcic vertisols, which indicate shrink-swell structures. And we see a very strange layer of stellate mud cracks. They are about 60 by 90 centimeters wide. And we actually see them at a lot of different sites as well. And this is moving towards the boundary beds. So it's actually quite nice that this actually seems to be a marker that we can use at different sites. And then we see an increase in what we call Catbergia burrows. We see Catbergia burrows actually aren't that prominent in the Catberg, but they have been named after the Catberg Formation, but they are invertebrate burrows, only about that wide in terms of burrows. They're little invertebrates, probably something like a shrimp-like creature that would have made them, but we see an increase in these burrows as we get towards the boundary beds. So what animals do we see? Well, we see the typical herbivorous dicynodonts. We see the animals that ate them. This is a Gorgonopsian. You can see why I say saber-tooth. It's got 2 canines coming out here. And I can finally instead of saying Gorgonopsian, actually give this creature a name. We've just published this year, and his name is Inostrancevia africana. And what is very nice is that this skull was found on this log in Spitzkop, and another skull and articulated skeleton was found on Loskop, and we're busy describing the postcrania of this animal now. So both at Noordgerdag, we have found 2 specimens of a new species. And we find typical Permian Therocophalian cerapsids like this little guy, and we find larger predators of Therocophalian cerapsids such as Mascarine, which we know survived the extinction. So what do the boundary beds look like? I've shown you this picture before. So we have these rhythmically bedded, these alternating dark reddish brown and olive gray mudstone, siltstone couplets. Now, you may have read in the literature, if your lecturers have forced you to read any literature on this, that in the older literature, the lime, the PTB, was placed at the bottom of these beds. This was when it was first discovered and first published on by Roger Smith and Peter Ward. But they, and eventually when I joined the team, were finding more Permian facies in these boundary beds and Permian taxa. They then placed it at the top of the beds. And really, it doesn't actually matter where the line is. At the moment, you could draw it in the middle if you wanted to. This is a transition zone from the Permian to the Triassic. And we find Permian taxa here, and we find Triassic taxa over here. So this is a PTB transition zone, and it varies in thickness. But this is what we look for, are these mudstone, siltstone couplets. And that is really what narrows down the area for the boundary. So above the boundary, we start to see an increase in rubification. That means reddening. So you can see the sediments here are redder, and we see these brown weathering nodules. We get very excited about that as paleontologists because we'll often find articulated skeletons in these. So this is what we look for when we're looking for fossils. We see the onset of gulling, and we see desiccation cracks. There are some mud cracks over here and ripple surfaces. And we start to see colloidal nodular conglomerate. So the taxa that we see, these are the 2 Lystrosaurus species that I've mentioned. These are the first taxa that we look for, and when we are looking for Triassic strata, and they occur about 3 meters or so above the boundary, and Proterosuchus as well. I think the lowest, not on this site, but at another site, the lowest I think has been 2 meters above the PT boundary on Bethel. And then we found some Therocophalians as well. This guy doesn't have a name yet. My PhD student, Brandon Stewart, is actually busy working on this Therocophalian. He's a new species, which is quite exciting because we don't find many new species in the early Triassic because we've studied it so much and found so many specimens. So it's really exciting that we found a new species in the earliest Triassic. So above this Kallenkloof member, which we consider to be Permo-Triassic, we find the Katberg Formation. And it is characterized, as you can see here, by these sandstone bodies that are parallel to one another. You can see them running here up the hill. And we consider the very first thickly bedded, laterally extensive, massive sandstone as the base of the Katberg Formation. We see increasingly thick sandstone bodies. We see a lot more colloidal nodular conglomerate. We see gulling and desiccation cracks. Here's a huge mud cracks over here. And just to point out here, this is an informal member, the Swartberg member. It's just a larger sandstone body. It's a regional member, which is why it has not been, it's not an official member, but it does help us to know kind of where we are in the Katberg, and we're in the upper part of the Katberg when we see the Swartberg member. So we've looked at the geochemistry of the site as well. And on the left, we find oxygen isotopes with an increase in temperature, which is to be expected. And on the right, we find a carbon 13 curve with a negative excursion just below the boundary. And these little squares are where we took samples, preliminary samples of from enamel, from Theriodont therapsids. And the geochemistry fits very similar to other sites in South Africa. Okay, but now to something that is a bit of a puzzle. And this is what I want to lead on to today. And we don't have an answer for this yet. So what we are looking at now are samples that I collected all the way through the sequence at this PT boundary site at Noordgedacht. So we are starting here in the Permian at the bottom of the site, and we are working all the way through, all the way higher, right up to the Triassic Catberg Formation. Now, I took samples of sandstone samples all the way through the sequence, and anything that looked interesting because we were looking for ash beds. Now, what we want to use in order to radiometrically date something is a pristine ash bed. We're looking for tuffs, and a tuff would be a huge amount of ash that comes from an active volcanic eruption from the time that you're wanting to date. And it is then put in the sediments, and you get a very, very high peak. You get many, many, many zircons, which are the little crystals that you date. And so you get a very clean ash bed, and you get a really good date with a very low error margin. Now, if you have other dates mixed in, so if you have some of these dates and you have older dates from older volcanic eruptions mixed in the sediments, you are going to have a curve that may look something like this, which is not really what you want. So what we did see is that we have a high peak at each of these samples, which we find is Permian to earliest Triassic in age. This is not a very concise age. This is something that you do to get a ballpark, to know vaguely where you are. LAISA PMS is much, much cheaper than radiometric dating, and you need to see where you are first before you continue to something that is going to break the piggy bank. So you can see curves such as this one with a high peak, and you can see there's very, very few other dates in this one. This is also another clean sample, but I did this with a lab in Stellenbosch, and they told me that none of these curves represented a tuff. And perhaps some of you in the audience or on Zoom could tell me whether you agree with this, if any of these curves represent a tuff, a pristine ash bed. So we don't really want to look at curves such as this because you can see there's a lot of other dates mixed in. This is what we'd call a detrital zircon sample. But so we did look at one sample. So this is detrital in nature. We got one large peak between 200 and 300 million years ago, but you can see there are some other dates, which is why this is not a tuff. Okay, this is what the zircons look like, the little crystals that we date. And we received a date of 251.7 million years, which is pretty good because the marine date is between 251.8 and 251.9. And our error margin is plus or minus 390,000 years. So it fits into this ballpark. So we know we are within the right region. However, with detrital samples, you get a very high margin of error. Plus minus 390,000 years is very large. And this date was found about 6 meters below where we put the boundary, and it's Triassic in age. But as I said, it's plus or minus nearly 400,000 years. We know we're in the ballpark. Somewhere where we have put the boundary is very similar to where the marine extinction is. So that date was published in 2020. There are newer dates coming, but I just wanted to show you that we do not identify the Perma-Triassic boundary in South Africa just on one thing. We look at a whole suite of characteristics that need to convince us that we are looking at a complete boundary sequence. So we will look at the disappearance of the Permian taxa, the appearance of Triassic taxa, the disappearance of the Glossopteris-dominated flora. We need to see a transition into the Dicrodium-dominated flora. We need to look for these mudstone, siltstone couplets. And if we've done geochemistry in the area, we need to look for a negative carbon-13 excursion. So there are many things that we look for to try and actually see where we are in a log when we are looking at a site. So some people have doubted whether there is aridity during the earliest Triassic. And this is to give you a little bit more geology again. We do see overall reddening. You can see this is my colleague Adam Huttenlocker sitting in a whole lot of reddish brown sediment, floodplain with claystone breccias. We see spheroidal glabules with septaria, sandfill desiccation cracks, and rough surfaced ferricrete and rhizocretions. We also have taphonomic evidence. Remember, this is the bone bed that I showed you earlier, and you can't see it as well here, but these are the ribs that I wanted to show you. So although these skeletons are disarticulated, there are some parts of the skeletons that have remained articulated, especially the ribs. Now, the ribs would only remain articulated in that way if skin had dried over those ribs in that way and kept them together, which means for some stage, these bones were lying there and drying up. So there was a dry spell. We also find complete skeletons, what we call mummified carcasses. This is Lystrosaurus here, and we can actually see skin impressions. So this animal died on the surface. It didn't die in a flood, and it just lay there and died, basically. And the skin dried over the carcass and mummified the skeleton. We also see skeletal weathering, and I can see this through the bone microstructure. Before these elements are even becoming fossilized, we can see cracking and evidence of weathering of the bones lying on the surface, being attacked by the sun and the wind and the aridity, the dryness, which you don't tend to see in the Permian. And there is also an increase in burrow casts. Now, burrows themselves are not evidence of aridity, but if you take a look at, for example, the Karoo Basin today, which is quite an arid area, lots and lots of animals burrow. And there are lots of reasons for burrowing, such as the protection of young. So it doesn't necessarily have anything to do with climate, but many creatures will burrow to escape extreme temperatures, something that's very cold, an area that's very hot, or an area that's very dry. So burrow casts do provide some protection against the elements, especially in harsh environmental conditions that we do see in the Triassic. Right, we see gulling at the base of sheet sandstones. So this is an example here. Vertically accreted, laterally extensive sandstone bodies, an upward increase in the thickness of multi-story channel sandstone bodies, and intraformational spheroidal nodular conglomerate filling these gullies. So what you have in here is a whole mixture of different pebbles and stones and bones. So these massive sandstone bodies are representative of a flash flood. So it's a massive dumping of sediment all at once, and it's aggressive and it's violent, and the sediments have been churned up, and if there are any skeletons in there, they have been churned up. We don't tend to find any articulated skeletons in this kind of conglomerate, but we will find smaller pieces of different things, which is why it's called a conglomerate. And so I think what researchers are finding, some of those that are finding the wet spells in the early Triassic may very well be sampling wet spells, because there were flooding, there were flash floods, but we can see there is definitive evidence of aridity. So there were times of drought and there were times of floods, but it was not seasonal. It was not predictable like we see in the Triassic. And this is the one big major thing, is the unpredictability. Animals can adapt to changes, but when it's unpredictable, it makes it very difficult for animals and plants to adapt to changes, because they don't know what's happening next. So it's the speed of change, something that's happening today, how fast the extinction is occurring, and it is unpredictability that is really the death of animals and plants. So returning to the flora, the plants, as I said, there are very few macrofossils, but if we have a look at pollen, we go back to Fielding's study. So Nowak said, no, he didn't think that there was much of an extinction. Fielding said, no, he thought there was, and that the extinction began some 400,000 years before the marine event. And then there was a peak extinction of the plants at the marine event. So if this is the case, if he's correct, that means that the plants began suffering about 400,000 years before the marine extinction, so that the plant extinction was more protracted. It was, it took longer. All right. So what we did is we then looked at the animals again, and my colleague Pierre Viglietti divided our logs and our data into little bins that we can look at it in ultra fine detail and look at the transitions of, here are the dicynodonts, here are the therapsids, the parareptiles, the diapsid reptiles, and the amphibians, to see the changes of who disappeared and who survived and who appeared after the extinction. And we found that on land, for the animals, the extinction lasted for about 1 million years. So it lasted much, much longer than the marine extinction. So if fielding is right, for the plants, it was about 400,000 years or so. For the animals, it may have been about 1 million years. So why the difference? I don't know. It may have been that the oceans offered some protection against changes in geochemistry and temperature for a while, and then just completely collapsed quickly. It may be that the animals and plants on land were more sensitive to changes in the atmosphere. I really don't know at this stage. So I'm just guessing at this stage, but this is what we are looking at the moment. So just to give you the very latest puzzle, what we are looking at the moment is that Bob went back to my site and he took a sample. This little line here, this olive gray line here, this is what I want to show you. Just ignore this right-hand side because it's not quite accurate. There's a bit of a mess in there. But I don't know what he was doing with the assemblage zones. But the important thing is that this is where we have traditionally placed the boundary. This is the Catberg Formation, which is Triassic. And this is where he took the sample, which is just below the Catberg Formation in the Pallincurve member. And he gained a date of 252.2. So this is a Permian date. And he said this was from a pristine ash bed. He said this was from a tuff. So it would seem that this is a very good date. So he did not perform PMS, but we did. I actually took that sample quite a few years ago, but being South African, RANs are a little bit difficult to come by. So I had not dated it with ID TIMS yet, but I did have a curve that I could actually see what his sample looked like. And it was this sample, which was actually pretty clean. Here's the very high curve, and there's very, very low any other dates, any older dates, but they are there. So my question to you out there is, does this represent tuff? Because the Stellenbosch lab said to me it didn't, but they may be incorrect. Does it represent a pristine ash bed, or are these still detrital zircons? And the reason I ask is very, very important, because this is where we place the boundary. Bob wants to place the boundary here at the base of the Catberg Formation. And so here is our older date with detrital zircons, 6 meters below the boundary with a Triassic date. Here is Bob's date, a Permian date just below the Catberg Formation. So we got some money together and we dated that exact same sample, and we got a Triassic date of 251.6 million years ago. Plus or minus 90,000 years. So the error compared to this date is much, much lower. And here is Bob's date. So now we have two dates, and one is Permian and one is Triassic. And they are very, very different. If you have a look at these errors here, you can see these dates are not going to overlap. There are two different dates. So what does this mean? And I don't know. I cannot answer this for you. This is for geologists and geochronologists to tell me what is going on. So does this suggest that there was reworking of the zircons in the sample, and that Bob did actually date a Permian zircons and we dated Triassic zircons, or does it mean something else? What is going on? We have that little seam that I showed you was on the one hill. We found it in exactly the same place on the other hill, and we've taken a sample and we want to date that. But if we do get a third date, can we trust it? Because it doesn't really matter if it's Permian or Triassic. We now already have two different dates for this supposedly pristine ash bed, so I don't know what we're meant to do with it. So what we are doing at the moment is, because I took all of these samples all the way through the sequence from Permian to Triassic, is we're taking the cleanest samples and we are dating them with IDT TIMS. So we are radiometrically dating them, and some of them are definitely not tough. Some of them may be, we're not sure, but we want to actually see a sequence. If we can start seeing a pattern then we might actually be able to figure out where we are. And does this line up with Bob's theory, or does this line up with where we traditionally place the Perma-Triassic boundary? In the end, though, I'm not sure if I really care, because I don't really work on the timing, the exact timing of the terrestrial extinction. I work on the biology of these vertebrates. So the fact that the terrestrial extinction was about 1 million years and the marine extinction was much shorter, does this mean that we are dealing with two different extinctions? And I don't think so. One was longer than the other, and the terrestrial extinction overlaps the marine extinction. So we are looking at the same extinction. One just took longer than the other one. And my interest here, well, as you can see here, there's a bit of a mess now, because now we have the detrital Triassic date in the Permian. Here we have two radiometric dates, which are detritals, but they show Permian dates. And here is Bob's Permian date, and here is our Triassic date. So it's a complete disaster. But my interest is finding out why these animals survived, who went survival strategies, because ultimately we need to look at today's extinction. Because for today's extinction, it is happening at a frighteningly alarming speed, not going to be able to adapt very well. And the global warming is very similar to what we see. The effects of how these animals and plants, particularly in the terrestrial realm, are being affected could have similar effects today. So we need to know what species are going to be most susceptible to extinctions. Thank you all for listening.
Jen, thank you so much. That was really, really interesting. Lots of cool questions that maybe we can help answer. So do we have any questions from the audience here in the room to begin with? And if you are watching on Zoom, you can simply raise your hand and we'll come to your questions later on, or you can just write them into the chat window as well. So we'll start with Sue. Sue, just loudly so they'll hear you on the microphone.
You see more extinction closer to that further away on a global scale.
No, not really. Although I must say that the other sites, which would be closer, so Russia also has really good records in terms of tetrapod preservation, but their biostratigraphy is not very well constrained. So it has been very tricky to actually know where you are in the Permian strata that do contain the Perma-Triassic boundary. So it's a little bit tricky to actually give you a complete answer, because people have tried, such as Mike Benton and colleagues, to really have a go at the Russian strata. But the biostratigraphy is not as well preserved. There are bits missing, and there's time missing there, definitely. So, but there are extinctions, and then there are appearances of new taxa. But in terms of the extent of the extinction, we see extinctions in the China terrestrial sections as well, which is much further north in China, closer to the Siberian traps. In terms of quantifying the extent, I don't think anybody has actually ever done that. But we need a lot more intensive study at those sites to compare it with South Africa, because we've looked at this so intensely. We need to take out that bias of collection bias, etc., with our sites here in South Africa.
Any other questions in the room? Okay, then let's.
Sorry.
No, sorry.
It's about the certain bone beds, and maybe there's any research on the behavior of this. So it was those bone beds caused by maybe transport by so they can perform inside of the.
So we do think that Lystrosaurus was social, because we do find lots of Lystrosaurus together. In that particular case with that bone bed, because there is disarticulation, we do think that those individuals were moved. So they died and disarticulation took place. And so there must have been some transport, some water transport, perhaps into a gully area or what have you. But then there was a drying out period, and that is where the rest of the bones all just fell apart. But that is where I'm suggesting perhaps where the ribs remained articulated, that skin was keeping parts of the skeleton skeletons together. But we often find Lystrosaurus in pairs. So Lystrosaurus burrowed, we have found many Lystrosaurus in fossilized burrows, and sometimes we find them in pairs, or we'll find them in pairs just where they died on the surface. And we find juvenile pairs, so we've even found two babies together. So they were definitely they weren't alone. They may have been in herds. We're not quite sure. That is a bit of speculation in terms of social behavior, but they were definitely at least in pairs, if not in larger herds.
Okay, and I think Reese has a question, then we'll come to Lou.
From a geophysicist, ignorance. You mentioned that chlorine and fluorine would impact ozone.
That's
I
what I've read.
presume
Yeah.
ozone or depletion thereof would have a different effect on land versus the ocean.
I would think so, yes. So
So is the rate of recovery of the impact of fluorine in the atmosphere known? And the chemistry of the oceans, so change in acidity, for example,
Yeah.
related to the
Yeah.
carbon emissions. Would that have a long-lasting effect? Do we know what longevity that would have?
we don't know, but that's a very interesting question. So if the land taxa are not as protected in terms of the UV ray for radiation, for example, could that have an effect? And yes, a lot of research has gone into acidification, acidification of the oceans and all of the geochemistry of the oceans. So, you know, there was a massive impact, but it seemed to happen very quickly. So if this extinction was happening already on land, and at times with the volcanic eruptions, with the Siberian traps, which it does, it was already happening, and then the marine biota just crashed. And I don't know why. And I don't think anybody else knows why either, because we've only recently figured out that the land extinction was taking longer than the marine one. So now we need to go and have a look at options and see, you know, what could have played a role in making these differences. Yeah, but that's interesting. Thank you.
Okay, Lou, we should be able to speak now.
Okay, thank you. I like your title, Apocalypse Then, which of course is a allusion to the cult movie Apocalypse Now, in which Robert Ball famously uttered the words, "I love the smell of napalm in the morning." And I tried to think of a catchy line like that you could apply to your fossil stuff, but I couldn't come up with something. But I'm sure you'll figure it out. So anyway, I have a comment and a question. The comment is, if you're strapped for cash to purchase radiometric dating by whatever method, I wonder if you maybe should consider using the arrangement we have with the Michael Wiedenbeck's lab in Potsdam, Germany, where he has a beautiful SIMS machine, secondary ion mass spec, basically an ion probe. And the arrangement we have is that he doesn't charge anything to people from Wits or from other institutions in South Africa.
Okay, that's fantastic.
Consider that. You'll get a lot of beautiful, very precise dates, and you know, it could save you some money for doing something else.
Yes, absolutely. Thank you.
Okay, so my question is, I guess I was expecting to hear more about what it is about these life forms, plants and animals, that allow them to make it through the extinction event, whatever it is, and as opposed to wiping them out completely. I was expecting some kind of bone story or a cell story or something like that. So can you comment on that, please?
Yeah, there seem to be several different factors that were important. Firstly, large taxa tend to go extinct. They're some of the first taxa that go extinct, and larger species perhaps just because they have to eat more to keep themselves going, they go extinct typically first. So it's not great to be large during an extinction, but we also found that in the early Triassic taxa, there are many, many taxa that burrowed. So burrowing definitely played some kind of survival role, either in keeping them away from extreme temperatures or in protecting their young. There were very few carnivores around in the earliest Triassic, so perhaps young protection was not that big a deal. So it was perhaps more about keeping away from harsh environmental conditions, either from the heat of the day or the dryness or extreme cold at night or something like that. But burrowing really did play an important role. And then what we are seeing with the animals that survived the extinction, we see this drastic change in life history where animals were growing normally and from juvenile to adulthood, dying of old age. Everything was rainbows and puppies in the Permian, and it was just a normal scenario. And this complete and utter change in the earliest Triassic with this unpredictable, harsh environment, this massive height in mortality rates, that these animals were dying young. So any animals that were able to breed young could survive. So we see a change in the reproductive timing. We see the presence of animals that were burrowing. We see smaller animals. And so that's factors like that. I don't think it's just one thing. For example, Lystrosaurus was both breeding young and we know it burrowed. It was a medium-sized creature, so it wasn't very, very small. But in terms of the other taxa, a lot of the early Triassic taxa were small little insectivores. So I think there are several factors, and you're basically pre-adapted to some kind of, to this kind of environment. And you're either luckily, pre-adapted, perhaps you're a generalist, you're not reliant on one particular plant species, for example, because if that plant species goes extinct in the Permian, you're tickets. So generalist species do tend to survive better than specialist species. So yes, there are several different factors that are involved in allowing these animals to survive an extinction.
And I guess on that note, there's a comment, maybe more than a question, from Johannes, who says maybe Lystrosaurus was nocturnal. They avoided harsh daytime radiation.
Maybe.
Yeah.
Okay. Any more questions from the Zoom audience or from the room? Yes, question over there. Louder.
But I was wondering, like, when it comes to mention how there's been a lot of study in period and the other side they haven't been as much. But it's like a maybe a bridge kind of study, maybe where they could like put the data points that exist instead of waiting for like volunteer studying to happen in places. If it's possible to put the existing data points to like probability distribution kind, where like South Africa has study more observation than more observation than as they observation and get it in
Well, we can try to match up the strata to see or the timing at least at different sites to see what are we getting at the different sites. So let's just take, for example, South Africa, Russia, and China. But there are a lot of gaps, so it's difficult to overlap the data. Also, because yes, you're right, we have to do some kind of probability distribution there because South Africa is so interesting study. We've got really good data there, but we don't have as good data. But recently, an American group went to China to look at the terrestrial boundary sites there, and we have recently obtained some better data of that site. So hopefully that's going to be coming out soon, and we can actually start making comparisons between different sites. It's been very difficult going to these Chinese sites particularly the terrestrial one, especially from outside of China. So it has taken a long time to be able to get to these sites. But I think they're coming out now, and but in terms of radiometrically dating these terrestrial sites, what many Chinese researchers have done is that they have tried comparing the Chinese terrestrial and marine sites with each other, and also very recently they have tried comparing the sites using mercury. So you also sometimes get a mercury spike at the Perma-Triassic boundary, and you see that in these Chinese sites. And we went out with a team and we didn't find a spike at Noordgebouw, but we did find a spike at 2 other sites in South Africa at that PT boundary bed where we put it. So so there are other ways of trying to compare, trying to actually match them, but yes, it's difficult and there are a lot of variables involved. But there are also good things. Lots of students have it.
Okay, any other questions? Yes, 2 more. Cameron and then.
It's just a comment. I'm also not a big part of so I'm seeing a similar issue now with the foreign is that our first like that, so flying the signal of that not going to be able to see the true currents of whatever. The other thing, there's one story as well is is there of important ages that you are reporting on ages, whether you're reporting youngest grade or youngest population or youngest population in there, there's the error margins on that as well. So you should things around. But and then also as well any sort of classification of observers. That age a bit more room, more before age or what. This is why it's good to have a good picture of knowledge to thank you for that once.
Well, we now have 2, so geochronologists. We have Roland Mandel and Jim Carley from the States. So we now have 2 geochronologists that are now working with us to try and Roland actually worked with us on the first date, but we now have Jim as well from Boise State University working with us, trying to figure out, how best to figure this problem out. And yes, we're reporting the youngest grains, but what Pia is actually doing now is she's trying to look at so what we're getting is we're getting averages when we look at when we plot our normal plots. But she's looking at Bayesian age models. Don't ask me how they work. This is new to me, and it's a new method. But we want to see what we're seeing through the sequence, and if that gives a better, accurate, true age of these different samples through the sequence, and to see what's happening there. And this is going to be part of our next research.
Okay, and the last question.
My question is not as technical, so I've had a good question about it and talking about social media. When you're watching movies and you need to
How do they know what they sound like? So in some cases, they can make some guesstimates. So, for example, Parasaurolophus, for example, is a dinosaur. I'll describe him to you. He's got this funny head dressing where it's a dinosaur with this bone that actually comes out the back. Do you know what I'm talking about? Okay, so they can actually look at the skull, and they can actually they can blow through different areas of the skull, and they can actually hear the noises that come out. It's very rare in that they can actually do that, and they can get a as I say, a guesstimate. It's not going to be the exact sound, but in some cases they can do that. But in other cases, no, they don't really know what the animals sounded like. They'll go on modern animals or modern reptiles, and if we are looking at movies, they're going to sound as scary as possible. So yeah.
Okay, thank you, everyone, especially for the questions, and another round of applause. And thank you, Jen, most especially. It was fantastic. Great to see so many people from the ESI. These talks happen every week, so please keep on joining us. And great to see so many students as well. Keep it in. Thanks, everyone.